Hello, I'm Ann Montgomery, Editor-in-Chief of BioProcess International Magazine. I'm here in Oakland, California this week at the BPI West Conference and Exposition. A number of speakers have agreed to share their insights with us. I hope you enjoy their comments and find them valuable as you approach your work.
My name is Susan Dana-Jones. I'm a Vice President and Principal Consultant with BioProcess Technology Consultants. I've been in the biotechnology industry for over 25 years, and I've been working with BPTC for over 13 years. I've worked both in the discovery and the early development areas. My specialties are cell line development, early process development, and the transition of those activities into production of early stage clinical material. So one of the emerging paradigms in the industry is the tendency for discovery and development to actually be interacting with each other. And this is new. It used to be discovery would run through a screening funnel, come up with one candidate, and hand it over to development. The change is that there's communication between the two functions such that the discovery function is given input from development to enable them to pick lead molecules that have better properties, that are easier to manufacture, that are more stable in solution, and that in the long run will benefit patients who will be getting better molecules that are more stable. So multiplexing, or bringing multiple candidates across from discovery into the early development activities, is a great way to reduce the risk of development because it allows you to actually start testing your molecules in early development activities in cell line development. There is a cost to that, however. There's a cost to each of the early development activities for a single molecule, and when you multiply that by bringing 2, 3, or 5, or 6 candidates into those early activities, there's an increased cost. I have seen companies willing to make that expenditure, understanding that it will give them better molecules and reduce the risk. There have been a number of talks here at the BPI West conference focusing on that very concept, that bringing multiple candidates into early development activities decreases risks, improves manufacturability, and really, on the long run, saves time. In the short run, it doesn't save time or money. So a key benefit to investors and patients of a multiplexing approach and bringing multiple candidates forward. In the long run, the key benefit is you have better molecules, you have better cost of goods, you have more stable goods so that they will have a longer shelf life. That gives you more flexibility when a product is commercialized, both in the manufacturing and the distribution, the storage in the hospital or the patient setting, and it benefits the patients by having, again, better, more stable molecules. Some companies don't want to try the integrated approach, but it is the emerging paradigm. There have been a number of companies presenting that very paradigm. Large companies, small companies, and virtual companies are adapting it. The roadblocks are certainly financial. Smaller companies that are not as well financed may not be able to take multiple candidates forward, or they're institutional. Large companies where they really have very different divisions of the discovery function and development function, maybe even in different locations, are less likely to be able to efficiently make that transition from the traditional silo approach to the more integrated and multiplexed approach. So there have been numerous technological advances over the last decade or 2 decades that have improved development, first of all. We have ways of imaging cell lines, so we can see when we have a monoclonal cell line. We have microbioreactors, such as the TAP-AMBER system, that allows you to test cell clones and then cell culture conditions in very micro scale, but mimicking what you would do in large scale. We have on the downstream side miniaturization and robots that allow you to do more screening of more column conditions rapidly to develop the processes. Those advances, which were made by development groups to facilitate development and shorten those timelines, have also enabled the multiplexing approach because it allows you, again, to bring more candidates forward, get them through some of the early activities, and come out with a process and a product that is superior.